Hello, welcome back to the second installment of our Chapter 8 discussion on recombinant DNA technology. This mini video is going to cover the tools and techniques that are used in the processes of trying to recombine genes and traits of different organisms to produce some beneficial outcomes, those three goals that we talked about in the first video. All right, so let's jump in with my notes here. The first one that's on the list is mutagens. So we saw that a little bit in chapter 7. So seeing how we can manipulate bacteria with mutagens to look for traits that we want to get. It's a little bit tedious and time consuming because you have no control over what the mutagens are going to do. You just are looking for mutations that then hopefully one is the one that you were looking for. So mutagens are a tool of recombinant DNA technology. The other one that's on the notes, or the next one on the notes, is called reverse transcriptase. We haven't talked about that yet. We will be talking about that more in chapter 13, since reverse transcriptase was first identified and comes from viruses. So as the name implies, this enzyme actually goes from an RNA molecule back to a DNA molecule. That is not something that normally happens in eukaryotic or even prokaryotic cells. It's our central dogma, DNA to RNA to protein. Nowhere in our cells do we go from RNA to DNA. That just doesn't happen. But in a particular group of viruses called retroviruses, they have their genetic information is RNA. And for that to work inside of their host cell, like a human cell, they have to be able to get from that RNA code into a DNA code. So what they do is the viruses bring in with them, when they infect the cell, this enzyme reverse transcriptase, and it makes a DNA copy of the viral genome and inserts that DNA into the host chromosome. And now you got viral genes in your DNA, and then they continue the replication cycle. For recombinant DNA technology, what they're able to do is we can find RNA a lot easier than we can the particular gene. Because remember, the gene is located in the DNA molecules with 100 of other genes, thousands of other genes. But if we have a little RNA transcript for just that particular gene of interest, we don't have to go searching for the gene. We can just get that mRNA, utilize reverse transcriptase, and make ourselves a DNA copy of that mRNA sequence. So this is kind of what happens. So you start with your mRNA, and this is just a picture from another source. It's got a little bit of extra details on it, but you start with the mRNA, you add a little primer to it, that's kind of these T's here, and then the reverse transcriptase makes a DNA copy, so that's the red, right? So this is the DNA version reading off of the RNA version, so that's the reverse transcriptase piece. So now we have the DNA. So now we have a half of a DNA. So then all we have to do is use DNA polymerase, which our cells normally have. It's easy to find that enzyme. And then it reads the complementary DNA to make a double-stranded DNA, which came from a single-stranded RNA, the mRNA that codes for the gene. So now we have a DNA gene that codes for the mRNA transcript, which can then be translated. So it's kind of you're backtracking. It's like reverse central dogma that researchers are able to use this reverse transcriptase to find to make a DNA gene that goes along with an mRNA transcript that is a lot easier to find than the actual gene. All right, the next tool is synthetic nucleic acids. So we know nucleic acids are made up of repeating subunits of nucleotides, right? So A's, T's, C's, and G's, the sugar, the phosphate, the base. And so before this technology was around, the only thing that could make nucleic acids were cells. All cells come from pre-existing cells, so that's where the DNA comes from. But as knowledge was increasing and technology was increasing, they were able to create machines, and that's a picture of one, a nucleic acid synthesizer, is you can put in your ingredients. I'm going to put in a cup full of A's and a cup full of T's and some G's and some C's and some enzymes and some ATP and whatever is needed to build your molecule, and then you just put in your sequence that you desire. So I'm going to put A, T, C, G, 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 A, T, you know, whatever you want, and then the machine will create a synthetic piece of nucleic acid for you, whether it's going to be a DNA or an RNA. The machine has the ability to make like a T, C, G, A, T, T, you know, whatever you need, it puts it in that order, and now you have a piece of nucleic acid with your specific sequence in it. Now, how are you going to know what sequence you want to put in there? Well, there's lots of different ways. We could get if you know a protein, you know the amino acid sequence, you could work backwards and figure out the mRNA sequence and find the complementary to that. So you could make your own gene that would eventually lead to that protein. You could make short little probes. So if you know the particular sequence, so let's say what I just wrote up here, TCGAAA, if you want to find that particular region on a gene, then you would produce a synthetic nucleic acid that would have that sequence and maybe have it be tagged with radioactive or fluorescent marker. And then you will be able to put that in your mix and it will go and complementary bind to that region of interest. So that's what probes are. And synthesizing antisense DNA, which is kind of the same thing, but we can use those for medical clinical applications. We'll see that when we get to antibiotics, is some antibiotics work by putting in your body antisense DNA that is complementary to the RNA, which blocks transcription or blocks translation. So synthetic nucleic acids, that machine is probably multi-thousands of dollars. I don't know how much it costs, but it's probably really expensive. The tool that's been used extensively in this kind of eruption of recombinant DNA technology from the kind of the 90s on is restriction enzymes. The newest kind of twist of restriction enzymes is CRISPR-Cas9 system. It's not been incorporated into your textbook quite as much as the restriction enzymes because it's still fairly new technology, but it works the same way, a little bit more specialized. But restriction enzymes were actually derived from bacteria. Same with the CRISPR-Cas9 mechanism. So these are enzymes that are already existing in biological systems. We have just kind of taken them to use them for our purposes. So what a restriction enzyme does is it has the ability to identify a particular sequence of letters on a DNA molecule, and it cuts the DNA at that particular sequence. It was thought that in nature, the restriction enzymes were used by bacteria to protect themselves from viral infection. So we talked a little bit about bacteriophages in transduction from chapter 7, that if a virus is inserting its viral DNA or RNA, if you can, as a bacteria, cut that up into little pieces before it has a chance to continue on with its infection, then you've just protected yourself. So that's the idea of where these restriction enzymes come from. So there's a couple different ways that they work, but both ways they identify what's called a palindrome. It's like that inverted repeat, like with a transposon. So here we have the restriction site, GAATTC on the top, and on the bottom reading in the other direction, GAATTC. So that particular sequence, GAATTC. An enzyme, they don't name this one, it'll just kind of cruise along the DNA, and wherever it finds that particular sequence, it will break the bond between the G and the A on both sides, and it will break all of the hydrogen bonds between the bases, like this. So right where that yellow line is, that's where the enzyme goes in and breaks those bonds. So it breaks the phosphodiester bond between the G and the A, and then it breaks the hydrogen bonds between the A's and the T's. Yeah, just the A's and the T's on that particular one. So as a researcher, if you want to cut out your gene, like we saw at the very beginning of the first video, that insulin gene that we want to take out of the human chromosome and place it into the bacterial plasmid, we can find these restriction sites on either side of the human insulin gene, cut it out, cut the plasmid with the same enzyme. So now you have these overhangs, are called sticky ends of an AATT, and then they'll just kind of, you put in some enzyme ligase and they'll fuse together, and you've just inserted your gene of interest into a bacterial plasmid or a bacterial chromosome. So sticky ends work, they kind of create this little jagged edge like this. Blunt-ended cuts do the same thing. They're looking for a palindrome. So in this first one, it's CCC-GGG, and then GTT-AAC on the green one. But when they find this particular palindrome, instead of cutting hydrogen bonds, they just split right down the middle, just like this. I'll erase these lines because that kind of is making it look like a sticky end. So there's our blunt-ended cuts. And so what they do, instead of having an overhang, they're just straight. And so you could use different restriction enzymes and use your ligase and seal those together. So you can actually seal the greens and the purples, even though you use two different enzymes, because they had the blunt edges, they were able to fit together. With the sticky end, you have to have matching. You'd have to use the same enzyme. So the next slide here shows where these enzymes come from, like what organism are we finding these from and what their restriction sites are. So the name of the enzyme is typically named after the organism that they find it from and how many they have. So EcoR1 is the first restriction enzyme that was isolated from E. coli. And then we have EcoR2. It was the second restriction enzyme that was isolated from E. coli. Or we have HIND1, or no, this is HIND2 and HIND3. And so if you take a look, we can see some of those restriction sites. I think I'm going to get a brighter color. The yellow is a little bit not very bright. So this is the BAM-H1, sticky, sticky. Oh, the EcoR1 was the one that we just saw in the previous example. This EcoR2 is a blunt-ended, blunt, sticky, sticky, blunt, blunt, blunt. So just a variety of different palindromes, these different restriction sites. And then if you'll notice, some of these don't have the actual letters. They have the abbreviations for purine or pyrimidine, they have some variation. So it doesn't have to be a particular letter. It could be one of the two letters that are the purines or the pyrimidines. All right, so those are our restriction enzymes. The next tool that researchers use to deliver their gene of interest to their target destination are what we call vectors. So vectors could be lots of different things. We saw one already with the bacteriophage in transduction. So it was the vector to deliver some of that bacterial DNA to a new recipient host. Plasmids are very common vectors in that we can put our gene of interest into the plasmid and then allow the plasmid to carry that into the cell. So in this particular example, we are starting with the mRNA for human growth hormones. That's going to be a protein. So we've been able to isolate the mRNA. We're going to use reverse transcriptase to make a copy, a DNA copy of this gene. And then we are going to use restriction enzymes on both the plasmid and the gene. So here's our gene of interest, human growth hormone right in the middle with a sticky-ended cut on either side, which is going to match perfectly here and perfectly here in our plasmid. And now the plasmid is going to get taken up into the bacterial cell. We then are going to grow the bacteria and harvest the human growth hormone. But you're like, well, there's an antibiotic resistance gene on that plasmid. What's going on? So an important characteristic of vectors is you need to know if your recombination was successful. So if you just put that human growth hormone gene into a plasmid without this antibiotic resistance gene and you plated them out, all the bacterial colonies that you would have grown, you would not have known who was actually the transformed cells and who are the normal cells. So then it would take a lot of extra testing to figure out which ones were actually successful in their recombination. So these are called markers. So vectors, good vectors are going to be small in size, they're going to be easily taken up by the host cell, and they have to have some kind of marker to let you know that the transformation was successful or the recombination was successful. So in this particular example, they just say antibiotic resistance. So they're not saying which particular one. So after we've made our gene We've inserted it into the plasmid. We put the plasmid with the bacteria in hopes they would take it up. We plate it out on a petri dish that contains that antibiotic. We'll just say penicillin. So the plasmid contains penicillin resistance, and then we tried to squish that human growth hormone gene in there. And so when we plate out our transformed bacteria, the ones that can grow in the presence of the penicillin are the ones that we know have been successfully recombined. And so then those are the ones that we can do research on, further research, or grow them in a big tank, and then you can then isolate your human growth hormone, and that's your product you're looking for. So those are vectors. Okay. The next tool, or tool of recombinant DNA technology are what are called gene libraries. So let's say you have a bacteria of interest. Let's say this is, we'll just say, what's a good one? Mycobacterium tuberculosis, right? So you're looking to do some research on tuberculosis. But it's a really hard bacteria to grow, right? It's got the mycolic acid, it grows really slowly, and it's really difficult if you wanted to do research on just maybe some of the genes that it has, not the whole organism. So what you can do as a researcher is you can take out the genome, you can cut it up with all these different restriction enzymes, isolating the genes of the genome. So in this particular organism, it only has 11 genes. That's not realistic, but you know. So then what you can do with your isolated genes, insert them into plasmids, which serve as a vector, and then you put each one of those plasmids into a completely different cell. So maybe these are E. coli. E. coli is really easy to grow. It grows really quickly, like 20 to 30 minutes for a generation time. So if you are a researcher studying tuberculosis, but you're only interested in this one particular gene, instead of growing Mycobacterium tuberculosis really, really slowly to get that gene, you could order from a company a culture of E. coli that has a plasmid that contains the first gene of interest of Mycobacterium tuberculosis, but growing in E. coli. So then you can do your research on that gene inside E. coli. So these are what are called gene libraries. So it's just a way to work with a particular gene of interest, usually grown in a different organism. Okay, so those are the tools that some researchers use. Now we're going to take a look at a couple techniques, processes that they actually do. The first one is called polymerase chain reaction, or PCR for short. It is basically an out-of-cell version of binary fission, where in a little machine, this guy right here, it's called a thermocycler. I found a picture of one so you can kind of see and envision what's happening. They probably cost about $10,000, so they're not super cheap to purchase. You might find some cheaper ones that aren't quite as accurate, like for classrooms and stuff. So you start off with your original molecule of DNA, and by the end of one cycle, you'd have 2 molecules of DNA. So you start with 1 and you get 2. And then if you take those 2 and you double those again, after the end of the second cycle, you will get 4 molecules of DNA. And the third cycle you double, and the fourth cycle you'll double. So in every cycle, you are doubling your DNA. So this is an exponential growth. So 2 to the n, just like we saw in chapter 6 with how to calculate bacterial population. It's the same thing, but instead of doing it in cells, we're doing it inside a machine called the thermocycler. So, and the reason why it's called a thermocycler is because it cycles through different temperatures, because the DNA and the enzyme activity is going to react to these different temperatures. So let's say we start off with the original molecule of DNA, your sample. You put that in, your thermocycler increases to 94 degrees Celsius. That's really, really hot. And what that does is it splits apart the DNA. And into this tube with your DNA, you're going to add some primers, DNA primers, you're going to add some nucleotides, and you're going to add DNA polymerase, right? So we're putting all of the necessary ingredients into this machine, and now we've separated the DNA. Now in a real cell, helicase does that. So remember, helicase is the enzyme that unzips the DNA. Well, we can't recreate that, but heating it up to 94 degrees does. So then when you cool it down to the next temperature, 65 degrees, what that does is it kind of lets everything settle where it's supposed to be. So your primers are going to be primed, your DNA polymerase is going to land on that primer, And then all of your nucleotides are floating around, and then you warm it up just a tad to 72 degrees Celsius. And what that does is it allows the enzyme to work properly, and it cruises right along, making a complementary copy of your original double-stranded DNA. So here we kind of started with dark purple and light purple, right? Comes from here. And now our copies are the green version. And then if we copy those, our dark purple gets a new green, the light purple gets a new green, and each green gets a new green. So these are all the new copies coming from the original. Now, this is used in research a lot because what it allows you to do is if you have a small sample of DNA, it allows you to make many, many copies of the DNA for your research. It's also really good for like crime scene stuff. If you get a tiny drop of blood and that's the only biological sample that you have, you don't want to run a ton of tests on it because then you're going to ruin that one sample. But if you can take a little bit of that small sample and run it through PCR, now you have a big whole bunch of DNA that you can do DNA fingerprinting and other types of analysis, and you're not going to destroy your entirety of your sample. So PCR increases the volume of DNA that you have, which allows you to do your research and your experiments a little bit better. So that's your PCR. Okay, the next one is gel electrophoresis. So gel electrophoresis, if you took by 211 here at RCC, you may have done this activity. It's usually the last one that we do if we have time in the lab. What it does is it allows you to take a couple pieces of DNA, cut them up with restriction enzymes, display them out on the gel, and your goal is to look for similarities in the sizes of the pieces of DNA that are cut, right? So let's say I have a piece of DNA here, and I have a piece of DNA here. And then I'm going to cut it with the same restriction enzymes, and I'm going to get those pieces, right? So I'm going to have 1, 2, 3, 4, 5, 6 pieces of DNA of the green version. And then with the same restriction enzymes, the blue gets cut like this. So I have 1, 2, 3, 4, 5, 6. I have also 6 pieces of DNA on the blue chromosome, but are they going to be of the same size? So if I try to mark it, so if those are my individual pieces of my blue chromosome or my green chromosome, and these are my pieces of the blue chromosome, I have 1, 2, 3 really small pieces of DNA on my blue chromosome and really only 1 small piece of the green chromosome. So they're not going to match up. So let's say we take our cut pieces of DNA, we put them in these wells, right? So there's my blue DNA, there's my green DNA, and then we use electricity, so we have a negative charge here and a positive charge. DNA is already negatively charged, so it moves away from the negative side, moves towards the positive side, and then it separates out these bands. So the green DNA moves this way, this far, if you will. The blue DNA, in this case, only had 2 pieces. And then what you can do is you take a picture of that, and you can then compare your samples of DNA. The more closely their banding patterns are, the more similar those pieces of DNA are. The more different those banding patterns are, the more different they are. So in our actual picture, this blue DNA is very different than what was cut with the green DNA. So they're not anywhere close to being related. Okay, and you may have seen these in what we call DNA fingerprints, are the actual pictures of those gel electrophoresis plates. And I have one that's kind of half cut off on my head, trying to see. So these 2 rows have almost the exact same pattern right across as I'm looking. This one is kind of like it, but that top bar is a little bit higher. But if you'll notice, it's not the same as that one. And these ones are similar. This one is kind of like this one. So those are the comparisons that we're trying to make. And so that's what gel electrophoresis does. And this is what we call DNA fingerprinting. So if you watch like your crime shows and they say, your suspect matches the DNA of your evidence, 99.999% chance that he's the one or she's the one, this is what they're kind of doing. They're looking at these similarities in these DNA fingerprints. Okay. And then the last thing kind of goes along with the gel electrophoresis. It's just a way to make a permanent record of your specific DNA coming from the gel electrophoresis. So we cut everything up, use restriction enzymes, cut things up, run the gel. But in the southern blot process is we actually stain the gel with some radioactive probes, and we put a piece of paper over that, like a photographic paper that will be exposed to the radioactivity, and then you basically develop a permanent record of your pattern of your gel electrophoresis. And it's called Southern blot because the guy who invented it, his last name was actually Southern. So it has nothing to do with the directions of like the compass. But interestingly enough, after he developed this process, other researchers kind of followed on that pattern. And so there's a Western blot and a Northern blot, and those were not named after people whose last name were Western or Northern. They were just kind of building off of this guy's last name was Southern. Western blot, I think, does proteins, and Northern blot does RNA, and the Southern blot does DNA. So it's a different molecule that they're tracking with their gel electrophoresis and the images that they're producing, but it's kind of the same process. All right, so I think that's it for our tools and techniques of Chapter 8. I hope you enjoyed it, and I will see you next time. Bye.